Okay, so thinking back to class on Tuesday, we were working through the kind of structural makeup of our skeletal muscles. We learned that skeletal muscle can be then subdivided into smaller structures. Okay, here on the previous page, you can see the whole muscle and opened up. You can see we have these bundle structures called fascicles or muscle fascicle as it's labeled there in the illustration. A fascicle is a bundle of what we call muscle fibers, and muscle fibers is just a fancy way to describe our muscle cells. And you open up a muscle fiber, you're going to find that you find what we call myofibrils inside. Myofibrils I describe as the contractile organelles. Okay, they're kind of rod-like structures, so they kind of have that tube shape that a lot of these other parts that a muscle has, whether that's the actual muscle fiber itself, the fascicle, or even in many cases the muscle, which is pretty tube shaped. Okay, this last slide that we left off talking about just kind of detailed muscle fibers and the fancy parts of them, which include those myofibrils, but also kind of some of these newer terms to describe things that we've already previously learned about, like the plasma membrane of a muscle fiber, which goes by the term sarcolemma. Okay, the sarcoplasm, which is the fancy term for the cytoplasm of a muscle fiber, and then some newer things like the T-tubes, which are these blue tube-like structures that carry action potentials inward, and then surrounding the T-tubes and surrounding the myofibrils in gold here is what we call the sarcoplasmic reticulum, or SR for short, which is just the same thing as the smooth endoplasmic reticulum, the SER, in non-muscular cells. So that's kind of where we left off. If I can go back down to this image that I told you at the end of class the other day, I wanted you to know, you can see inside of myofibrils, there are smaller structural parts that I'm going to talk about now, what we call filaments and sarcomeres. Okay, so before I start talking about on that page, I have the kind of flow chart that I created there last time, brought it with me from a while ago, and now I'm going to finish filling it in and adding those two structures. Find a pen. Here we go. So we saw at the end of last class what we call myofibrils. I kind of just described them as contractile organelles. Okay. Structures smaller than myofibrils inside our muscles are what we call sarcomeres. Okay, and a sarcomere can be basically just described as, sorry, it's not letting me focus. They're the functional units of a myofibril. And inside of sarcomeres are what we call filaments. Sometimes you'll see them listed as myofilaments. Myo is a prefix that always means muscle. Okay, so that's what myofilament translates to, mean muscle filaments. Okay. And that's going to be where I leave off the kind of flow chart. Okay. Now, if you want to kind of imagine what a sarcomere is, okay, so just, I'm going to do an awful drawing. I know I'm not a good artist, but hopefully this will help make some sense of it. Okay, a single myofibril, remember, it's kind of a tube-like structure. So here's my version of a myofibril. There's my best drawing of a three-dimensional tube-like structure. Okay, within myofibrils are individual segments. okay, a segment of a myofibril. So from like here to here, that's what a sarcomere is. Okay. And inside of the myofibrils will be these, or excuse me, inside of the sarcomeres will be these filaments that we'll talk about. And I drew them kind of like that, because that's really, if I go back to the lecture, kind of what it looks like. If I go back here, you can kind of see the little lines. Okay, the thick and thin filaments are labeled. Those are the two major filaments, okay, that are found inside the sarcomeres. Here is one sarcomere illustrated. Okay. Down at the bottom of this next page, we've got a myofibril, at least a segment of it. Okay. And we can see we've labeled one sarcomere. Okay. If this was a longer image, it'd be going in this direction and this direction. Then there'd be another sarcomere there. There'd be another sarcomere there. Okay. So let's just read through and explain what sarcomeres and filaments are. Within myofibers are smaller structures called filaments. The two major filaments are the thick and thin, named because one's thicker than the other. Simple as that. The thin filaments are about 8 nanometers in diameter. They're about 1 to 2 microns in length. Thick filaments are about twice as thick as the thin filaments. They're 16 nanometers in diameter, but they're about the same length, 1 to 2 microns. Okay. Both the thick and thin filaments are what are directly involved in the contractile process. So they are going to be what allow for contraction to occur. The amount of overlap that is found between these filaments depends on whether the muscle is in a contracted, relaxed, or stretched state. We'll kind of talk a little bit more and see an illustration kind of demonstrate that here in the next slide or two. Filaments inside the myofibril do not actually extend the entire length of the muscle fiber, but instead are arranged in compartments called sarcomeres, which, as I wrote on the other page, are the basic functional units of a myofibril. Okay, sarcomeres are lined up in series and include the following kind of regions or parts to them. And that's what the kind of two-part illustration showing you. This is a nice three-dimensional illustration, and this is a more two-dimensional illustration showing you basically the same things. Okay, we'll first start by talking about what we call the Z-discs. Okay, Z-discs form the border of a sarcomere. So in other words, a sarcomere goes from one Z-disc on one side to a Z-disc on the other side. So that's why we can see here where they've labeled it, the sarcomere runs from the Z-disc to Z-disc. So what is a sarcomere, or excuse me, what is a Z-disc? They are a type of dense protein material that serves as the place of attachment for the thin filaments. And if you look in the two different illustrations, okay, here in this top illustration, the real thin red lines represent the thin filaments. Okay, down here in this picture, the yellow lines represent the thin filaments. And notice in both cases, the thin filament is directly attached to the Z-disc. It's kind of easy if you think like Z-discs, the way they're drawn, they're like zigzagged. So Z for zigzag is kind of an easy way to remember Z-discs. Okay. Now, we talked the other day about the fact that cardiac muscle and skeletal muscle is striated, meaning it has those light dark bands repeating arrangement. And if I go back, to this slide, we can see those both here in skeletal and cardiac muscle. Okay, it's because within the sarcomere there are segments where there are only thin filaments. There's segments where there's only thick filaments, and then there's segments where they overlap. Okay, we call these different regions like zones, bands. Okay, and so let's first talk about what we call the A band. Okay, the A band is the darker middle part of a sarcomere, and it extends the entire length of what we call the thick filaments. So in other words, where you can see the thick filament on this side, where you can see the thick filaments on this side, from here to here is what we call the A band. Okay, A band down here illustrated thick to thick. Okay, that's going to produce those dark bands or dark striations. Okay, whereas the I band, we can see there's two of them, one on each side of the sarcomere. Okay, I bands are going to be where there's only the thin filaments, where there is no overlap with the thick filaments. Okay, and actually an I band spans across two sarcomeres. So the Z disc, which borders the sarcomeres at the end here, okay, but the I band actually extends across into a neighboring sarcomere. So you can see where the other sarcomere will be started going in this direction. The other sarcomere will be going in this direction. Okay. An I band spans across two different sarcomeres. In this case, we can really only see a little bit of the two neighboring ones. Okay. These are going to be why you see the light striations. So it actually goes light, dark, light, dark, light, dark, which is nice because I is in the word light, A is in the word dark. So that's kind of an easy way to remember it. Okay. Now, going back to the A band, let's talk about what we call the H zone or H band, as well as what we call the zone of overlap. Okay. The H zone or H band is the segment of the A band where there is only the thick filament. Okay. In other words, there is no thin in the A band, but in the two zones of overlap, it's exactly what its name implies. This thick and the thin filaments are overlapping each other, okay, and thus are telling you exactly what their name means, where the two filaments are overlapping. Okay. Okay, so once again, pattern of thin, thick filament overlapping, consisting of zones of bands, creates striations, can be seen in single myofibrils and in the whole muscle fibers. So it's this, that is the reason why skeletal muscle, and we see the same thing in cardiac muscle because they have sarcomeres and thick and thin filaments, all that wonderful stuff. That's why they look striated under a microscope. Okay. As far as this kind of two-part image, I'm going to tell you right now, it will be on your exam here next week. Okay. So put a big fat star in it, highlight it, whatever you need to do, make sure you know it. Won't be one or two questions. There's probably going to be several questions related to this. You'll have to be able to label the different parts of it. So it's a very, very important image. Okay. Now, in parentheses here behind the words thin filament, we see the terms myosin, actin. There's another filament I haven't mentioned called titin. Okay. Those are proteins, and we're going to talk about these proteins right now. There are three types of proteins that help form and build our myofibrils. At the top here, contractile are the two that were mentioned on the previous page in parentheses, myosin and actin. Then you've got regulatory proteins and then structural proteins. Okay, so let's talk first about the two contractile proteins, myosin and actin. Those are the ones that actually generate the force during contraction. So the actin and myosin that are mentioned here on the previous page are what actually do the contracting. We'll start by talking about myosin. Okay, here's a couple pictures to illustrate what myosin is. Basically, you can kind of think of an individual myosin, which is what we show here, as being like two golf clubs, where the shaft of the golf club, the long part of the golf club, is twisted around each other. Okay, and then the two heads are shown here at the end, just like in a golf club. If you don't know what a golf club looks like, okay, I'm sure everybody does, but the head of a golf club is what you actually hit the golf ball with, okay, are shown here. Now, it doesn't really do it justice, but these two heads are kind of pointing in directions about 60 degrees away from one another, okay? So they're not pointing directly in the same direction. They're pointed just a little bit. Think of them like they create a V between the two of them. Associated with both heads are what we call actin binding sites and ATP binding sites, which do exactly what their name implies. When ATP binds to the binding site, it gives energy that then allows it to connect and pull on actin when it binds to actin. Okay. Now, what myosin does is each of these actin binding sites is able to connect to an actin on actins, what we call myosin binding sites, and that forms what we call a cross bridge, a connection between the thick and thin filaments. Okay. Now, unlike a golf club, which is a rigid structure, and that by that I mean like the head, it stays in that flexed position. In the case of these two myosin heads, they have a hinge point, which is kind of like your elbow. which, as we know, our elbow can flex and extend. We can see that the head is in two positions, or these two heads are in two different positions. One's more of an extended position, one's more in a flex position. Okay. Before attaching to an actin molecule, what the heads need to do is get into this extended position. And then that way, when they attach to the actin, okay, they're going to be like grabbing a rope and pulling it to yourself. And if you want to grab a rope and pull it towards yourself, you want to have your arm extended when you grab the rope. That way you can pull the rope farther. If you grab the rope with your arm already bent, can you pull it anywhere closer to your body? Not really. Okay. So that's what the hinge is important for. It enables it to go from a kind of a cocked or flex position, shown here, to more of an extended position. Okay. And what's really cool is these two heads kind of just alternate and the grabbing and pulling of actin. Just like if you're pulling a rope with two hands, you grab and pull with one hand, then you grab and pull with the other hand, and you break it free and grab with the other hand again, and you keep pulling and pulling and pulling. That's what these guys do. Okay. Now this is one individual myosin. Okay. Here's what one thick filament looks like. A thick filament is strictly made up of myosin molecules. Okay. In fact, there are about 300 myosin molecules that form one singular thick filament. Okay. And if you look hard enough, you can kind of see right here would be the M line, which I forgot to mention on the previous page, is the middle part of the sarcomere. M for middle is the easy way to remember that. Okay. But going in one direction from that M line to one side are myosin molecules, and then going in the other direction from this point to the other would be myosin molecules pointing towards that other Z disc. Okay. Now notice we can see the double heads to all those individual myosins, and what that is going to enable is for a three-dimensional thick filament to be formed. Okay. If I go back to this picture, if I look at this segment of the figure that's more three-dimensional, see all these big red dots? Those are thick filaments, and there's heads kind of pointing in all sorts of different directions. And what's nice is surrounding each thick filament, you see all these little yellow dots. Those are the thin filaments. Okay. And let's talk about the thin filaments now. Okay. Actin is the main component of the thin filament. It's not the only component. Okay. Myosin is the only component of the thick filament. Actin is just one of three. Okay. But it's the main component. That's why on the previous page, behind thin filament, you see actin in parentheses. Okay. Here's what actin looks like. Okay. There's actually two forms: G-actin, F-actin. It's short for globular and filamentous. Not super important that you know that. But think of like each of those little G-actins like a bead. Okay. And you take beads together and you can make a beaded necklace. That's what F-actin is. More like a necklace, except it's really kind of two strands coiled. Kind of looks like DNA in that it's got that helix shape to them where they're kind of twisted the way that they are. Okay. Each of those G-actins has a myosin binding site. And then when you put it in together, forming the strands, notice that the myosin binding sites are going to be lined up the way that they are. Okay. Now, the other two parts to a thin filament are the two regulatory proteins, tropomyosin and troponin. Okay. Regulatory proteins are important for switching the contraction process on and off. Okay. When put together, the three proteins, actin, the main component, plus tropomyosin and troponin, they form the thin filament. So there's three proteins that form a thin filament, only one that forms a thick, and that's myosin. Okay. Now, in this case, in this situation, this illustrates a thin filament that's not ready for contraction. Okay. And that's because this kind of rope-like protein, tropomyosin, it's covering up all those binding sites where myosin can grab and attach, and grab and pull on actin. Okay. But if the muscle is releasing calcium, remember calcium we learned is stored in the sarcoplasmic reticulum. That calcium comes out and it's going to bind to these troponin molecules, And that causes the tropomyosin just to kind of move out of the way. It just kind of shifts it out of the way. Those binding sites get exposed, and then the myosin heads can grab and start pulling on the actin. Okay. And as the grabbing and pulling occurs, what happens is the thin filaments get pulled towards the M line, towards the middle of the sarcomere. And so the thick filaments end up getting pulled towards the Z discs. And this is what happens. Okay, look at those different bands that we talked about, the H band, the I band, the A band. Okay, well, the H band, remember, that's the part of the sarcomere where there's only thick filament. I band is where there's only thin, and A band is just thick to thick, which includes the H band and the two zones of overlap. As we go from a relaxed state to a partially contracted, look what happens to the size of the H band and the I band. Okay, the H band and the I band are getting smaller because those thin filaments are getting pulled towards the M line. The Z discs are coming closer, the sarcomere is getting shorter. What doesn't change, and we can really appreciate that down here when the muscle is maximally contracted, is the A band. Okay, because when a muscle contracts, the filaments don't get shorter, they just increase their length of overlap. Okay, the way that I always kind of just demonstrate this is by using my arms. So I'm going to move my camera off real quick. Put my camera on, I should say, not my computer screen. So I'll stand up. Okay. So let's take my right arm. You know, we'll say this is the thick filament, this is the thin filament. It's not perfect. Okay, but right now I got my arms like this with just a little bit of overlap between the two. Okay, we pretend this is a Z disc, this is a Z disc. When the muscle goes from relaxed, it starts to contract. Look what happens to the zone of overlap. The amount of overlap increases. Okay, the length from elbow to elbow is now shorter, so my sarcomere is shortened. Have the lengths of my arms changed? No. Okay, so the filaments don't get shorter, they slide over one another. Okay, and that's why the description of muscle contraction is described as the sliding filament theory. And we don't get into that in this class, it's physiology based stuff. So for those of you who are gonna, or are in physiology, you'll have that to look forward to here in a few weeks. Here's what it looks like for real in some microscopic views. We can see all those different bands and zones labeled. You can see the Z discs really nicely on the ends. We can see how it's light, then it's dark, then it's light. Okay, as we go from relaxed to contracted, the A band never changes. Okay, the I band and the H band or H zone definitely do though. And that's because the zone of overlap has grown tremendously. Now, there is one final type of protein that makes up our myofibrils, and they're structural. And that's because they help just kind of keep structure together. They keep the thick and thin filaments in the proper alignment, give the myofibril elasticity, extensibility, and link myofibrils to the sarcolemma and extracellular matrix. Okay. The first one, which is the third most plentiful protein in our entire skeletal muscle, is titin, and that's what was actually shown here on the previous page. Kind of looks like a coil or a slinky. Okay. Titin gets its name for its really big size. That's what titin means. Okay. But its job, and I would highlight this, is to stabilize the position of myosin by spinning from a Z disc to an M line. So basically, it runs from the Z disc all the way to the M line and just helps hold the thick filaments in place. Okay. I would also highlight that this accounts for much of the elasticity and extensibility of myofibrils, which we talked about those terms the other day. We have seen those before as well. Elasticity, think of that as like the ability to recoil or to go back to its original shape after stretching, and extensibility is the actual stretch. Okay. Dystrophin is another structural protein, links thin filaments of the sarcomere to integral membrane proteins in the sarcolemma. You guys ever heard of muscular dystrophy before? Anybody ever heard of that? Okay, it's called muscular dystrophy because the most prominent form of that, there's a few different types, but Duchenne's, I believe is how you pronounce that, muscular dystrophy, is a congenital condition that results in improper forms of dystrophin. Unfortunately, there's no cure, there's no way to prevent it. It basically leads to muscles just breaking down and eventually the person doesn't survive as a result. So really, really crappy thing. Lifespan for people with muscular dystrophy is not very long either. And then finally, Nebulin, which wraps around the entire length of each thin filament, helps anchor the thin filaments to Z discs. As far as the different images on this page, don't worry about them, just the information. And then looking here on the next slide, basically it's the exact same kind of flow chart that I created for you guys. I just wanted to do it as we ran through. Table 10.3 just gives you those different levels, pictures, and description. The only difference is they don't have a little segment between myofibrils and filaments for sarcomeres, and that's because sarcomeres aren't actually a structure. They're just a part of a structure. They're part of a myofibril. So we had, what, 6 things listed. They've got 5. So, okay, very last thing, touching back on those other 2 types of muscle really quickly. Let's talk a little bit about each with regards to cardiac and smooth. Okay, cardiac muscle has the exact same arrangement of actin and myosin with the same band, zones, Z discs as what we see in skeletal muscle fibers. That's why it is striated. Okay. Other parts of a cardiac muscle fiber that are important to talk about are what we call intercalated discs. That's what these little zigzag dark bands here are. Okay. They're unique to cardiac muscle fibers, so we don't see them in anything else. And we've talked a little bit about that already. What they are irregular transverse thickenings of the sarcolemma connecting the ends of muscle fibers with one another. So in other words, it's just a thickened plasma membrane between the 2 cells. Okay. Here is where we're going to find some special features that hold those cells really tightly together, but also you're going to find what we call gap junctions, which are special openings within an intercalated disc, allowing our action potentials to spread from one muscle fiber to another. Okay. And that enables all those muscle fibers to basically contract at the same time, something that's very important to control your heart rate and your heartbeat, and all that stuff that I really don't get into in this class. As far as the 3 connected tissues that we talked about last time, cardiac muscle tissue has 2 of them, endomysium, perimysium, but there is no epi. Okay. When stimulated by an action potential, cardiac muscle tissue remains contracted about 10 to 15 times longer than what we saw or see in skeletal muscle tissue due to a longer delivery of calcium in the sarcoplasm. Mitochondria and cardiac muscle fibers are more numerous and are larger, suggesting that cardiac muscle fibers to make their ATP aerobically, which means with oxygen. And like skeletal muscle, cardiac muscle fibers can undergo hypertrophy, which we talked about that term the other day, which means that the fibers can grow in size. And this is going to explain why many athletes actually have an enlarged heart. You see it a lot in like distance runners, swimmers, bicyclists that are doing, more of that aerobic long-term type exercise. But they call it an athlete's heart for that reason. Okay. Down on the bottom right, we've got a couple of illustrations talking about or illustrating smooth muscle, which there's 2 forms, visceral or single unit, and then multi-unit. Okay. The difference between the 2 really relates to how nerve signals travel into and through the cells. Okay. The more common of the 2 is visceral smooth muscle or single units. We find this in the skin. Tubular arrangements form parts of the walls of the small arteries and veins, and hollow organs like our stomach, intestines, uterus, and urinary bladder. Okay. In the case of these guys, they're going to have gap junctions just like what we see here. So what that means is the neurons, the nerve cells that stimulate them, a branch doesn't have to connect directly to each cell, okay? Because when the nerve signal comes to like this cell or this cell, there are those gap junctions, those doorways that allow the action potential to spread from cell to cell to cell. Whereas with these over here, multi-unit, we can see each cell has to have a direct connection to a neuron, okay? So they don't have those gap junctions connecting, okay? We find multi-unit in the walls of larger arteries, airways to the lungs, the rector pili muscles, which we learned previously help make your hair stand up and give you goosebumps. Muscle of the iris that adjusts the pupil diameter, so it's in your eye, as well as in the ciliary body that adjusts the focus of the lens in your eye. Okay. Over on the right, they show that we don't have that same arrangement of thick and thin filaments in smooth muscle, although they are there. They're just not in those orderly circumere arrangements as what we see in the other two. So I would highlight that phrase there. Smooth muscle does contain thick and thin filaments, but not arranged orderly in circumares like we see in cardiac skeletal. We also find what we call intermediate filaments, which are not found in the other two types, but I'm not even going to really get into what that means or anything like that nature. As far as, you know, the pictures of this page, don't worry about them either. So just a little bit of information on those two types of muscle. And with that, we'll call it a day. And it's about a week too.